Dr. Orwig, thank you so much for being here.
Thank you, Miranda, for the kind introduction. I'm going to see if I can share my slide with you now. Okay, are you all able to hear me and see my slide?
Yeah.
Yes.
Okay. Well, so first of all, thank you for the kind introduction. This, of course, is my first time visiting the ALD conference, and it's been a great opportunity for me to learn a little bit about the disease. I direct the fertility preservation program in Pittsburgh that lives in a new center that we opened actually right at the beginning of COVID in March of 2020, called the Center for Reproduction and Transplantation. So we cryopreserve tissues and eggs and sperm and embryos for patients who are at risk of infertility for a variety of reasons, including medical treatments that they might receive. And we have, in fact, encountered families with ALD. We have not yet cryopreserved a tissue for a kid with ALD. That family decided not to go forward. But I think that what I share with you today related to other kinds of patients can easily be applied to ALD patients as well, and it'll give you an idea about what options are available. And if we cryopreserve tissue for a young ALD patient, how those tissues might be used in the future for reproduction. So I will, after giving myself a pointer, I'll start out with just a brief introduction about the process of spermatogenesis. And I think what's important for this audience to understand is that spermatogenesis is a stem cell-based system. There are stem cells in the testis that produce sperm throughout the post-pubertal life of all men. So it's very much similar to the way the bone marrow works, that there's bone marrow stem cells that continually produce blood cells and immune cells throughout life. And I assume that you are largely familiar with bone marrow transplant because that's one of the primary treatments for ALD. So the stem cells that are in the testis and produce sperm are called spermatogonial stem cells, or SSCs. And these cells function like every other stem cell type that I know about, and that is that they self-renew to maintain the stem cell pool in the testis, and they also differentiate to produce a progenitor that gives rise to the entire spermatogenic lineage that ultimately results in the production of these terminally differentiated sperm that have a tail so they can swim through the female reproductive tract, and they carry the 23 chromosomes from dad into the uterus to fertilize the egg. And this process of spermatogenesis happens within the seminiferous tubules of the testis. So testes are made up of a bunch of seminiferous tubules that look like spaghetti. And every one of these seminiferous tubules both begins and ends in this structure here called the reedy testis. So sperm are produced in the tubule, they flow out to the reedy testis space, and then into the epididymis, where eventually they come out into the ejaculate. And the reason I'm taking just a minute to show you about the anatomy of the testis is because it's actually this space here, the reedy testis space, that we target when we want to transplant testicular stem cells to restore sperm production in a male who is infertile. This is a technique that's experimental. It's not ever been done yet in the human clinic, but progress in animal models are very promising, and we believe that this will be a technique that we're able to offer to patients in the future. Well, if this process of spermatogenesis works normally, then a man should produce in his ejaculate a total of about 40 million sperm. So guys produce tons of sperm. But we know this doesn't happen in the normal way all the time. And in fact, 1% of people in the global population have a condition called Asospermia, which means they aren't producing any sperm. And one of those circumstances that can cause Asospermia are chemotherapy or radiation treatments for cancer or for conditioning prior to bone marrow transplant, which is something that you guys would be familiar with. So because we know that medical treatments such as chemotherapy and radiation can cause permanent infertility, we should advise all patients or their families about the reproductive consequences of the treatment that they're going to receive, and also about options for preserving fertility. So for adult men and women, those options are standard of care, and they include cryopreserving eggs or cryopreserving sperm or cryopreserving embryos, because we know that these cells can be thawed in the future and used with existing assisted reproductive technologies to achieve a pregnancy and have biologically related offspring. But unfortunately, those options are not available to prepubertal boys or girls, and that's simply because they're not yet producing mature eggs or mature sperm. So using standard of care technologies, we don't have any way to preserve the fertility of kids that are getting these medical treatments that could cause infertility. And at least as it pertains to cancer, the consequence is, well, the good news is that the 5-year survival rate from cancer is now over 85%. So we're producing lots of survivors who still will have their entire reproductive life in front of them. I assume that that may also be true for ALD patients that are getting bone marrow transplants that will essentially cure their disease. So what we need to understand is that even those prepubertal boys who aren't producing sperm, they do have those spermatogonial stem cells in their testes. They're just sitting there waiting for puberty to happen. And then once puberty happens, they'll begin producing sperm. And it turns out that it doesn't matter whether you get a spermatogonial stem cells from a 1-year-old or a 5-year-old or a 10-year-old or an adult, those stem cells are equally capable to restore sperm production after transplantation. At least that's true in mice. And if the same thing holds true in human patients, then it would be reasonable to cryopreserve testicular tissue with stem cells, even for the youngest patients. So for that reason, and because we have several experimental methods in the research pipeline that we believe will be available in the future that will allow patients to use their frozen tissues to produce sperm and have children in the future. There are centers all over the world, including ours, that are actively freezing testicular tissue for boys and ovarian tissues for girls, because we believe that we'll have new technologies that will allow those patients to use their tissues in the future and to produce eggs or sperm. Well, we established our fertility preservation program here in Pittsburgh in 2010. It was at that time that we obtained approval to cryopreserve ovarian tissues and testicular tissues for patients who were at risk of infertility. And since that time, we have frozen testicular tissues for 486 boys and ovarian tissues for 169 girls or women. So the question is, if you have a patient or a child who's at risk of infertility due to their medical treatments, when should you refer for fertility preservation? And the answer is as soon as possible, but it should be before the medical treatment that would cause infertility. In the case of ALD, I think typically we're talking about a myeloablative chemotherapy that reduces the patient's own bone marrow stem cells to create space for the transplanted stem cells. So this discussion about fertility should happen before that preconditioning treatment and as early as possible, because that allows us time to schedule the surgical procedure, and even better, if we could combine that surgical procedure with another procedure that your child might be having to manage their disease and or to get ready for bone marrow transplantation. At least related to our program, it is easy to get in touch with us. We have dedicated both phone lines and email addresses that we can help you access these experimental fertility preservation options, either here in Pittsburgh or through one of our more than 30 collaborating centers around the United States. And I'll show you those in a few minutes. So when you call our fertility preservation line, we have a wonderful and multidisciplinary team that helps to coordinate the surgical procedures as well as the processing and cryopreserving of tissues. But the people that you would encounter immediately will be Brooke McClendon or Rachel Neely, who are our two program coordinators, and they take care of everything. So they'll schedule surgeries, they coordinate with the lab to process and freeze the tissue, they communicate back with the families both before the procedure and also after the procedure to let them know what happened. this is the team that comprises what I called our new Center for Reproduction and Transplantation that we opened in March of 2020. But we're proud of the fact that patients have traveled from all over the world to access these experimental fertility preservation options that we have here in Pittsburgh. That's because there really aren't very many places in the world that you can access these technologies. The number is not 0, but it's also not 100. So there are very few places where you could access these technologies. So we're proud of the fact that patients are coming from all over the world, but we also recognize that this may constitute a barrier in access to care, either because families have a sick child who's not able to travel, or because the cost of travel is prohibitive and they wouldn't be able to do it. So to start to break down that barrier in access to care, we developed a coordinated network of centers who all collaborate with us to provide fertility preservation options. And those are indicated by all of the circles on this map, and now numbers a little bit over 30 across the United States. I won't bother to tell you what all of the different colors mean, but what I will tell you is that with this model, a patient who was in Seattle, Washington could have the surgical procedure to collect a testicular biopsy in Seattle, and then the tissue would be express shipped to our center over here in Pittsburgh, where we have the infrastructure and the personnel to process and to cryopreserve those tissues for the family. But the family wouldn't have to travel to Pittsburgh necessarily to access these services. So just to give you an idea about the types of medical indications that lead families to request fertility preservation for kids, these are the type of diagnoses that we've seen in our center over the past 10 years. You can see that in kids, we see a lot of CNS tumors, sarcomas, and leukemias, but also we have over 20% of our patients are patients who are receiving bone marrow transplant for a non-cancer disease, and so ALD would fall into this category. And because of the toxicity of the treatments that are required for myeloablative conditioning, those patients we consider to be at significant risk of infertility, and therefore they're eligible for the fertility preservation services that we provide. When we receive a sample in Pittsburgh, regardless of whether it's from surgeries that are done locally here at the University of Pittsburgh Medical Center or from one of our collaborating centers around the country, those tissues are processed by simply cutting them up into small little pieces. These measure between 2 and 5 millimeters across. And then we use a controlled slow rate freezing process to slowly reduce the temperature to a very low temperature so that those tissues can be maintained in a frozen state for a long period of time. When we do this under a research protocol, and if it's in Pittsburgh, we cover the cost of the surgical procedure, as well as the cost of processing and freezing the tissue. 75% of the tissue is frozen for the patient's future use. 25% of the tissue goes to research so that we can develop technologies that we hope will be available and allow patients to come back and use their tissues in the future. I told you I wasn't going to tell you about what the colors on all those circles meant, but one of the things they mean, we provide these services in Pittsburgh through research protocols where we cover the cost that I told you about. We also can provide these services through a compensated mechanism, either in Pittsburgh or through one of our coordinating sites. In that case, 100% of the tissue would be stored for the patient's future use, and none of it would go to research. So just to review what I've told you so far, for males or people with testes who are going to get a medical treatment that might cause infertility, the standard of care option to preserve future fertility is to cryopreserve a semen sample with sperm, because we know that those sperm can be frozen and thawed in the future and used to achieve a pregnancy with existing reproductive technologies such as in vitro fertilization. For kids or other patients who aren't able to produce a semen sample with sperm, I've told you that we have the option to cryopreserve intact pieces of testicular tissue. I've also told you that we have a variety of methods that are in the research pipeline that we will believe will be available and allow us to use those tissues in the future to produce sperm. And I put red boxes around two technologies that I believe are mature and actually ready to translate to the clinic now. In fact, we're approved to do both of these technologies in our Center for Reproduction and Transplantation. So we're actively recruiting patients to do the first transplants. And these two technologies, one is a cell-based technology, and that is to transplant those spermatogonial stem cells directly into the testis in the place where spermatogenesis happens. And in that case, those cells will restore sperm production, just the same way that transplanted bone marrow stem cells restore blood and immune cells in the body. We have another method that's a tissue-based method, and that is we take those little pieces of tissue that I told you we cut the testis up into, and we just graft that tissue under the skin of the patient where it matures up to produce sperm, and then we can recover those sperm and fertilize using IVF or similar technologies. So I have a limited amount of time today. I'm just going to show you a little bit of the data that we've done in a monkey model where we've demonstrated that transplanted intact pieces of testicular tissue can produce sperm and offspring. So this was a study that was done by Adetunji Fayomi, who was a PhD student in the lab, and Karen Peters, who was a research associate, and she's actually now a nurse working on a cancer floor. So in a monkey model, we treated monkeys with a chemotherapy that we knew would cause permanent infertility. In fact, the treatment that we use is very similar to the myeloablative conditioning that many patients would get prior to bone marrow transplant, including either busulfan or busulfan in combination with cyclophosphamide, which may be some drugs that you all have heard about. Before we treated with chemotherapy, we took a biopsy from the testes of these animals and we cryopreserved those samples using that controlled slow rate freezing method, and then stored the tissues in liquid nitrogen. Then after the animal was treated with chemotherapy and made infertile, we thawed out the pieces of testicular tissue and put them back into the same animal. So the experimental design looks like this. As I said, we transplant the intact pieces of tissue. Here you can see 4 little pieces of tissue that we put underneath the skin flap, and then the skin flap is simply sewn shut. And we did this in 8 different locations. We did it in 6 locations under the back skin and 2 locations under the skin of the scrotum. And this experimental design allowed us to ask questions about graft outcomes or transplant outcomes from fresh tissues, fresh testicular tissues versus frozen and thawed testicular tissues. Of course, for our patients, we're most interested in frozen and thawed because if we cryopreserve tissues for a kid, those tissues may be frozen for many years before they come back to use them. This design also allowed us to ask questions about graft location under back skin versus in the scrotum. And also we introduced Matrigel into some of the sites. This is kind of a jelly that we hope would cause blood vessel development to occur and support the survival of the tissue. Well, it turns out that it didn't matter whether the tissues were frozen or whether they were fresh. It didn't matter whether or not we added that Matrigel that would cause blood vessel development. And it didn't matter whether the grafts were under the back skin or under the scrotal skin. The sample or the grafts grew continuously throughout the duration of the experiment. And you can see that after time, those teeny pieces of tissue eventually produced rather large lumps, either on the back skin or under the scrotal skin of the animal. So after about 8 to 12 months, we recovered those grafts. And you can see that those tiny pieces of tissue now have grown and fused together into one large mass. And when we dissected the tissue apart, we were able to release what were normal looking testicular sperm for a monkey. And it wasn't just a few sperm like you can see on this slide here. We were actually able to release between thousands and millions of sperm from every single graft that we recovered. So the next question for us was, are those sperm functionally able to fertilize an egg and establish a pregnancy? So to address that question, we sent these sperm to our friends at the Oregon National Primate Research Center in Portland, Oregon, because they have an in vitro fertilization lab for monkeys. And so they took the sperm that we sent to them and fertilized monkey eggs. And the result was that those sperm were able to fertilize, producing 2-cell embryos and 4-cell embryos, 8-cell embryos, and etc., all the way up to the stage of a hatching blastocyst. This is a stage when the embryo would normally implant into the uterus. So, having confirmed that the sperm could fertilize and produce a blastocyst stage embryo, the next step for us was to transfer those embryos into a surrogate female monkey. We did this in November of 2017. 1 month later, we were able to confirm that there was a biochemical pregnancy. 1 month after that, we could confirm on ultrasound that not only was the pregnancy still there, but the fetal development was going normally. So at that time, we programmed a cesarean section for full term of pregnancy. And the result was the birth of Grady, which stands for graft-derived baby. She was born on April 16th of 2018, and she's the first monkey baby who was born from autologously grafted. That means grafted back into the same patient from which the tissue came. Frozen and thawed prepubertal testicular tissue. Now, Grady still lives at the Oregon National Primate Research Center, which means I'm responsible for paying her bills until the rest of her life. So I'll get to watch her development. One of the neat things about her being in Oregon is that they have an established program for monitoring the development of these babies and comparing how Grady develops and what her behavior and social interactions are like compared with other monkeys who were born in the normal way. And by all indications, Grady's development is normal, just like any other monkey that was born in the normal way. And here you can just see one of the observation periods where the technicians are monitoring and quantifying behavior. Grady is shown here with the surrogate mother that carried her during her pregnancy. So just to sum up what I've told you today, the fertility preservation program in Pittsburgh provides a comprehensive menu of fertility preservation options, both for adults and children. And those include both standard of care options, egg freezing, sperm freezing, embryo freezing, but also experimental options such as testicular tissue and ovarian tissue freezing. It's important, though, to acknowledge that there are not yet any reported human live births from testicular tissue transplanted back into patients. This is research. As I said, we're approved to do that now, but we haven't done our first transplants. So it'll be a few years whether we know that this thing that I just showed you in monkeys also will be possible in human patients. But the value of the monkey experiments at least shows us that the methods are safe and that they're feasible and that we could get a healthy offspring who had normal development. And finally, we are happy to help anyone coordinate fertility preservation care, either here in Pittsburgh or through one of our coordinated sites around the country. We're happy to just discuss and consult either about the risks of infertility caused by the medical treatment and or about the options that might be available for each individual patient depending on their unique circumstances. And this phone number and email address are dedicated specifically to the program, and they're continuously monitored by our program coordinators. So with that, I'll finish up just by telling you that I've described to you a clinical program that we developed recently, but the research program is something that we've been working on for, 20 years of time. So there are lots of people to acknowledge, but I tried to acknowledge the people that specifically did the work as I went through the talk. We have collaborators all over the world, and really this is such a early stage technology that that's the best way we can move the field forward is by communicating with each other and sharing our ideas. And finally, I always like to finish up these talks by acknowledging patients, because that is who inspires the work that we do. But also in many cases, they donate tissues to our research so that we can develop technologies that we hope will be available for those patients in the future. So with that, I thank you for your attention, and I'm happy to answer any questions that you might have.
Thank you, Dr. Orwig. That was so interesting. I learned so much in a very short period of time. First, we need to know, does Grady have her own Instagram? I need to Grady.
Grady doesn't have her own Instagram, and unfortunately, I can't help her with that because I don't know anything about Instagram.
No, thank you for sharing that. This
Yeah,
research,
no problem.
it's someone said in the chat, it's just so hopeful, especially for us parents. Kathleen from ALD Connect, I had the same thought, but she worded the question much better. So for this 20% that's non-cancer, you said ALD would fall into that category. I would imagine there's other genetic diseases in there.
Yeah.
So her question was, when you release the sperm post-transplant, is it possible to select only the non-ALD Y chromosome sperm so that the concern of passing on the ALD is gone?
Yeah, so that's a really good question. So the answer is yes, you could do that. The way you would do it isn't actually by selecting sperm, but when you go to the IVF clinic, the female partner will be stimulated to produce eggs. Usually there will be several embryos that result after fertilization. The number might range from 2 to 10, but sometimes even can be higher than that. And those embryos can be sexed for Y chromosome. And so then you could transfer only the embryos that were the male embryos if you wanted to avoid, or sorry, the female embryos, if you wanted to avoid the possibility of passing it on.
Interesting. Okay, and then I had one other question about the storage. So the 75% going to storage, 25% going to research. I was wondering, is that kind of a part of the deal when you sign up for this? I personally believe in giving to get, but I know that, that could be kind of a gray ethical issue for some people. So how does that work for people who maybe?
Yeah, it's a really good question, because something that we battle all the time is barriers in access to care. I told you about one of them, which is travel and the cost of doing that. Cost is another barrier. So if we can reduce that cost, that's great. If not, the cost of doing the surgical procedure and processing could be a barrier to families. What we have strived to do is to create as many different avenues to access fertility preservation as possible. So the answer to your question is that if you were to come to Pittsburgh now and do testicular tissue freezing, it would be under a research protocol. In that case, we would cover the cost of surgery and processing and freezing the tissue, as well as the first year of storage, and 25% of tissue would go to research. We offer similar services through all of these sites around the country. Some of them are under a research protocol. In that case, the cost of surgery depends on the center, but we still cover the cost of processing and freezing, and 25% goes to research. But we ran into exactly the families that you talked about, or institutions. Some institutions have said we won't participate in that way, and some families have said that we don't want 25% of our child tissue going to research. So we created a new mechanism. It's a fee for service, where families can pay for the cost of surgery, processing, and freezing, and then 100% of the tissue we would save for their future use. None of it would go to research. Interestingly, after we established that mechanism, we realized we're able to provide that options for patients at 30 different sites around the country, but we couldn't do it at our own institution. So now we are in the process of applying for permission to make this available as a fee for service in Pittsburgh as well. So hopefully there's a lot of avenues to access the technology depending on what fits every individual family.
Thank you. All right, I'm just going to do one more question because it seems quick and it's a yes or no. For the surgical procedure, is the entire testicle removed or only a portion?
Yeah, so we have the option either to do a biopsy, which would basically be to take a pie-shaped wedge that constitutes about 25% of one testis, or to take a whole testis. But as you can imagine, families are sensitive to taking a whole testis, and so only a few out of nearly 500 families now, only a few have opted to take a whole testis. Most of them are getting a biopsy of one testis that constitutes about 25% of the testis.
Great. Dr. Orwig, thank you so much for your time today. We really appreciate learning about this.
You're welcome. Thank you for having me.